Hi, I'm Brian Malow, and I'm speaking with Maria Dzunkova, and she's a microbiologist at Berkeley Labs. Thanks for speaking with me, Maria.
Hi.
And you are one of the 12 finalists or semifinalists from the Berkeley Lab. You're one of the 12 finalists in the 4th annual Research SLAM. Congratulations.
Thank you.
Are you excited? Are you nervous?
Yes, I am excited, but I'm not nervous. I think that it's going to be a lot of fun.
Excellent. Have you done a lot of outreach in addition to your research?
Yes, of course. I love writing my scientific blogs when I'm trying to explain my research in a way that general public would understand it.
Excellent. And so you're at the Joint Genome Institute in the Biosciences Directorate, and you're a microbiologist. Did you always know you wanted to be a scientist? And did you always know you wanted to be a microbiologist?
Yes, I always knew that I wanted to be a scientist, but obviously when you're just like 18 years old, you don't even know what kind of research field will appear in the future. So when I was 18, so I started to study plant genetics because I wanted to be involved in plant breeding and help to get rid of famine in Africa. But it has changed with the time because I just got like a part-time job in a sequencing facility, and DNA sequencing just became like a very, very important field. And so at that moment, we were mostly sequencing genomes of smaller organisms, not big plants, but just small bacteria that have smaller genomes. And this was just the right time when I still had the possibility to switch from the field of plant research to the microbiology. So then I have worked in different places around the world, and I have learned a lot about microbes and the beauty which can be found in different ecosystems.
Excellent. Where are you from?
I'm from Slovakia.
And is that where you started as an undergrad and maybe
no,
what was your path?
Not at all.
Oh, no, not at all. Tell me about your path that led from Slovakia to Berkeley Lab.
So I went to university in Prague. So this used to be the capital of Czechoslovakia. This is something, like people from east of Slovakia have in mind that they need to go to some bigger city to get to the best university. So this is how I ended up in Prague. And there, so in Europe, we have exchange programs for students. So I went to Spain, and this is where I met my husband. And basically, I ended up doing my PhD there. I'm doing my PhD, so I went for a research stay to Harvard. So I kind of already knew how is research going on in the US. I like it a lot. And then I went for my first postdoc to Australia. And in Australia, my former boss, he used to be a very important scientist of the Joint Genome Institute. So this was like a very natural path that I went from Australia to JGI. So that's how I ended up here.
Excellent. And so what are you studying now? What are you going to be talking about for 3 minutes in your Research SLAM presentation?
So I will be talking about the microbiome of nudibranchs. Nudibranchs are colorful slugs they mostly live we can find them in tide pools. You can just drive 1 hour from the Berkeley Lab and you will find them if there is low tide. So this is very exciting. And I work in the single-cell genomics group, and we are always looking for some cutting-edge technologies to get very interesting results that you couldn't get with just normal technologies that everybody is using. So we were looking for some special fluorescent probes that could serve to enrich for some bacteria with some special features. And we were trying to find some bacteria that have the feature of synthesizing some secondary metabolites, which can protect them or their host against predators. Like they are usually like toxic, but if you use the right dose, they can be used for some maybe like protection of plants against insects, or also inhibit the growth of cancer cells. I don't know. This will be very, very long path in the future, but this is our starting point. So we used these fluorescent probes. We applied them to the microbiome of nudibranch, and we were just expecting to find like some subset of bacteria, which will be maybe different from the total composition. But what was interesting that we have found only one bacteria with this special feature. So, and this bacteria, interestingly, at the moment when I find it, so I was thinking that I was the first one to discover this bacteria that belongs to very, very strange kind of family of bacteria that nobody has discovered before. But during that time when I was working on that, so I have just found out that somebody in Australia and somebody in South Africa has already identified some other members of this big new family. But I still have my bacteria, which is very unique to nudibranchs, and it's interesting. And yeah, and it's producing something that protects nudibranchs from the predators.
That's fascinating. What's it like to be the first person to know something about the natural world?
This is very usual feeling actually in when you do metagenomics. So it means that you just sequence all the DNA which is in some sample, like some microbes that you collect from some environment. It's very, very common that you discover new bacteria or new viruses or even new kind of life forms that nobody has knew about before. But the thing is that we just discovered these sequences. So we don't physically have any picture, like we don't take pictures by microscope. But in this nudibranch project, we are going to have some microscope pictures because we are really focusing on this single bacterial species. But normally when we work with big DNA sequencing data, we discovered heaps and heaps and heaps of new bacterial species, and we just deposit them to the databases. And we don't really care about them, but they are there. So really, this is not like a feeling, oh, I have discovered something great. No, this is like everyday feeling. Like,
well, that's because you're a microbiologist. If you were studying large mammals, you wouldn't get to discover a new one very often. Totally. But you study microbes, and so there's so much left to be discovered. That's fantastic. And what gets you up in the morning to do the kind of work, and to the lab to do the kind of hard work you do? What's your motivation?
So I just love learning new things, and just the normal scientific curiosity that I think that everyone who works in science needs to have.
And this outreach part, so you like to do that and do the research, but the outreach, you said so you have a blog and you've done a lot of writing?
Yeah, not recently, but in the past, we used to do that. And I think it's very important because, you know what, so recently, you know, with the pandemic, so I just noticed that people just don't know what the scientists are doing. And maybe that's why like so many conspiracies are coming out. And people just don't know like how the scientific discussion looks like what we actually need to do to demonstrate something, and that we really need to start with the idea that we don't know like what will be our final result, and we have to be like we totally have to forget about our personal opinions, like I think that this will end up this way. No way, you can't do that. Like you have to be like totally objective and just like wait until you get your result, and then try to make the best interpretation of the results as you can. And if you cannot, so then you ask your colleagues who have better knowledge about it, and so on. So this is the process that I think that everybody needs to understand. So science is not a question about any opinion. This is the results, the facts that you get. So we need to inform the public that this is how the science is done.
Excellent. Well, thanks for taking the time to talk to me, and good luck in the research slam.
Thank you.